Hi, once again welcome back to Obstetrics and Gynecology channel. Today we are going to see about an important and an interesting topic, that is fertilization. So let us see what is fertilization. Fertilization is the process of fusion of the spermatozoon with the mature ovum. That is, fertilization is the fusion of ovum with the sperm, and we are getting the zygote, and the zygote will future develop into the baby, fetus, and finally we will be getting the baby. So first we should see how this ovum and spermatozoon or sperm is getting formed. So for that we should understand what is oogenesis and what is spermatogenesis. First we are going to see the oogenesis. Oogenesis is the development of mature ovum in the ovary. That process is called the oogenesis. In the ovary, we know that in the female body, it's a female reproductive organ. Already we have seen it in the anatomy of female reproductive system. If you are having any doubt about the anatomy, you can go through that. And these germ cells which are present in the ovary, it undergo the mitosis. Mitosis is a type of cell division which is occurring in the normal cells. So due to the mitotic division, we are getting the daughter cells of same chromosomal pattern and the same size and shape. Same type of cells will be getting after the mitosis. So these germ cells undergo the mitotic division and it forms a cell known as the oogonia. And this oogonia will mature again and it will form the primary oocyte. This primary oocyte, its chromosomal pattern is 46X. And this primary oocyte will undergo the first meiotic division. That is the type of cell division. Meiosis means it's the type of cell division which is occurring in the sex cells, and the chromosomal pattern will become half. The secondary oocyte, which is getting developed after, or which is getting formed after the first meiotic division, it contains 23 pair of chromosomes. 23 chromosomes, 23 X chromosomes, that is half of the chromosomes, and balance half of the chromosome is there with the first polar body. It is there in the first polar body, but all the other cell, cytoplasm, cell organelles will be with the secondary oocyte. The first polar body won't be having those things. So it's only a cell membrane or only membrane with the chromosome, that's all. But all the other cell organelles and everything is with the secondary oocyte. And this secondary oocyte is still in the ovary. Then in this form it will get released from the ovary, and that process is the ovulation. Normally we are telling that release of ovum from the ovary we are calling as ovulation, but actually the ovum is not getting released. The secondary oocyte is getting released from the ovary. And this secondary oocyte, if it is getting fertilized, that is, if it is getting union, or if the union of ovum and sperm is occurring, we know that it is occurring in the fallopian tube. If it is occurring from the fallopian tube, it will undergo the balance stages of division and multiplication that we will see separately. And if it is not getting fertilized, it will degenerate within 24 hours. It will get destructed within 24 hours. But once if it is getting fertilized, again it will undergo the second meiotic division. And after the second meiotic division, we will get the female pronucleus and the second polar body. This female pronucleus and second polar body will be there. So this female pronucleus will be developing into the future fetus, and this will be having the chromosome pattern of 46X. So balanced development of the fetus we will see after discussing about the development of the sperm. Already we have seen the development of the ovum and up to the process of fertilization. Union of sperm and ovum we have seen. So now let us see how the sperm is getting developed. The process of development of sperm is known as spermatogenesis. And in the male gonads, germ cells are there in the male gonads, and this also will undergo the mitosis, and after the mitotic division it will form the spermatogonia. This spermatogonia again mature and it will form primary oocyte. This primary oocyte will undergo the first meiotic division and it will form two daughter cells, 23X and 23Y. One daughter cell will be with the 23X and another daughter cell will be with the 23Y. But same here in the case of oogonia, we have seen only one daughter cell is forming, and along with that a polar body is getting formed. But here two daughter cells are forming, two sperms will be developing. And again this undergo the second meiotic division, and we will be getting 4 subsequent cells. 23x 2 cells will be there, and 23y 2 cells will be there, and these cells are known as, after the second meiotic division, the cells are known as the spermatids. These spermatids undergo a process known as spermiogenesis, and after the process of spermiogenesis we are calling the spermatids as spermatozoa. So that's all about the development of the sperm. And now let us see the structure of ovum. See, here we can see a nucleus is there in the center, and it is surrounded by the cytoplasm, and it's having a thicker proteinaceous membrane that is known as the zona pellucida, and cell membrane or vitelline membrane is there, and outside the vitelline membrane, follicles of corona radiata is there surrounding that. So if you are looking into the structure of ovum, its size is 130 microns, and it is the largest cell in the body, and its outer covering or cell membrane we are calling as vitelline membrane. Inside the cell membrane, a zona pellucida, outer transparent mucoprotein layer is there, thick mucoprotein layer is there, that is known as the zona pellucida. And the space between the perivitelline and zona pellucida is known as perivitelline space. Zona pellucida and that cell membrane or vitelline membrane is known as the perivitelline space. And the covering granulosa cells, this entire structure is covered by granulosa cells, and those granulosa cells are known as the corona radiata. And if you are looking into the structure of sperm, it's having a head, middle piece, and a tail. This head is having a cap-like structure. Here you can see a cap-like structure. This is known as the acrosome, and a nucleus is there in the head, and this is the centriole, and this is the mitochondrial axial filament. It's having head, neck, middle piece, and a principal piece. Head is derived from the nucleus, and it is covered by a cap-like structure called acrosome. Already we have seen in the picture, it is covered by a cap-like structure called the acrosome. And its neck is narrow, and it contains a funnel-shaped basal body and a spherical centriole, and it is called the connecting piece. Neck is known as a connecting piece. And axial filament, it is covered by a spiral sheath made up of cell mitochondria, and it helps for the movement of the sperm. At the end of the spiral sheath, there is a ring-like structure, and that ring-like structure is called the annulus, and the entire sperm is covered by a plasma membrane. Sperm capitation or acrosome reaction. Capitation means it's a physiochemical change in the sperm by which sperm become hypermotile and is able to bind and fertilize with the secondary oocyte. It takes 2 to 6 hours. Actually, in the head of the sperm, we have seen that it's having a cap-like structure known as the acrosome, and this acrosome will undergo certain physiochemical changes, and it gives motility for the sperm, and it helps the sperm to bind with the secondary oocyte or bind with the ovum. That process is known as the sperm capitation or acrosome reaction. That is, activation of the acrosomal membranes will occur, and due to the activation, these acrosomal membranes will release an enzyme known as hyaluronidase. This hyaluronidase will help the sperm to digest the thicker zona pellucida of the secondary oocyte. In the ovum structure, while we were discussing, we have seen that it is having corona radiata. Outer corona radiata is there, vitelline membrane is there, and inside the vitelline membrane, thickest mucoprotein zona pellucida is there. And actually the sperm is supposed to penetrate all these layers, especially the thicker zona pellucida. And this enzyme hyaluronidase is helping the sperm to penetrate that membrane. And later, this process is known as the acrosomal reaction. The sperm plasma membrane fuses with the acrosome membrane, and it binds with the zona protein also. And this sperm plasma membrane, acrosomal membrane, and the zona protein, these three things will bind together, and it will form a thorough binding or thorough covering against the fertilized ovum, and it prevents the polyspermy. That is, once if a sperm has entered into the secondary oocyte, immediately it will form this covering structure around that fertilized ovum, and it further it prevents the entry of sperms into the oocyte, secondary oocyte, or into that fertilized ovum. So only one sperm will be having the chance to enter into the ovum, and it prevents the polyspermy. Polyspermy means entry of multiple sperms. Next is the fusion of the gametes. Already we have seen it is fused. That process is the fertilization. So we have seen up to the fertilization, and I have told you that it will immediately after the fertilization, this secondary oocyte will undergo the second meiotic division. Immediately after that, it will go and it will happen for the secondary oocyte. Then fusion of the two nucleus will occur, and after that, that nucleus is known as the female pronucleus. This is the thing which is happening immediately after the fertilization. The two nucleuses will get fused together, and it will form the female pronucleus. And this process up to the formation of the female pronucleus, after that fertilization is occurring in the fallopian tube. Later it will move to the uterus, towards the uterus, and it will get implanted inside the uterine endometrium. That process is known as the implantation, or it is otherwise known as the nidation, and it will occur approximately 8 to 10 days after the fertilization. And some of the books are telling on the 11th day after the fertilization the implantation is occurring. The stages of implantation, or the process of getting implanted or nidation, includes 3, that is apposition, adhesion, penetration, and inversion. Apposition means the zygote reaches to the endometrium, that is it will come towards or near to the endometrium. And adhesion means it will just adhered or attached, it will get attached to the endometrium, that is the adhesion. Next is the penetration. It enters into the fold of endometrium, that is known as a penetration and inversion. That is, the type of implantation in the human being is interstitial implantation. Interstitial implantation means the fertilized cell or the zygote is getting implanted inside the endometrial cells. It will just erase the endometrium, and it will get entered into the endometrium, and finally it will be covered by the endometrial cells. That is, it is not growing inside the uterine cavity, it is growing inside the endometrial cells. That type of implantation is known as the interstitial implantation. Next we can see the development of the fertilized ovum. This ovum will undergo multiplication. Already I have told you that female pronucleus will form. It is in that stage. Now it will get multiplied, and it will form. It will come to a 16 cell stage. It will be looking like a mulberry, and in that stage it is known as morula. 16 cells will be there. So the morula will be like almost looking like this. Like this, 16 cells will be there. See, clumped cells together, 16 cells will be clumped together. Later what will happen means a cavity will, the cells will be tightly packed. There won't be a gap like this. It will be tightly packed. Okay. Later what will happen means a cavity will develop inside this here. See, if I'm just redefining, it will be like this. The cells will get segregated to one side, and outer membrane will be there. A cavity will develop. See here, you can see a cavity. See, this cavity will be there, and fluid will get filled into this cavity, and this time it is known as blastocyst. That is, these cells will just get differentiated, and it will go to the one corner, and a cavity will develop. In that stage, this ovum we are calling as, or zygote we are calling as, fertilized to that structure we are calling as blastocyst. And later this covering membrane, outer covering membrane will get differentiated, and that stage it is known as blastocyst. Now it is blastocyst. Blastocyst covered by a single layer of cell known as trophoblast. The remaining cells clump together at one end and it is forming the inner cell mass. Already I have drawn the previous picture. See, here the cells are clumped together. This is not this. Later this will form the inner cell mass, and this outer layer of the blastocyst, this outer covering of the blastocyst will be known as, this single layer is known as the trophoblast. Here also endometrium will be there, and here also endometrium will be there. And later this trophoblast, this outer covering of the blastocyst, that is already I told you that it will be named as trophoblast. This trophoblast will develop the placenta and the chorion, and this inner cell mass, the cells which are clubbed at the end or in one corner, it will develop the fetus and the amnion. And we have seen the cells which are segregated in one end, we are calling as this inner cell mass, and I told you that this inner cell mass will get developed into the fetus and the amnion. This inner cell mass, later it will get differentiated into three layers, that is ectoderm, mesoderm and endoderm. Ectoderm is the outer layer, mesoderm is the middle layer, and endoderm is the inner layer. This ectoderm forms the skin and nervous system of the baby, and mesoderm forms the bones, muscles, heart, blood vessels, including those of the placenta, blood vessels of the placenta, and endoderm forms the mucous membrane and the glands. Later, this ectoderm, mesoderm, endoderm, these three layers are there, and this is that cavity of the blastocyst. Already it was there, and it is covered by all these cells. Okay, ectoderm, endoderm, mesoderm. Later what will happen means, near to the ectoderm, towards the outer portion, this is the ectoderm, a cavity, another cavity will develop, and the cells of the ectoderm these cells will start to get differentiated, and it will produce a fluid known as the amniotic fluid. And this cavity, initially it will be there only at the top, later it will develop and it will cover the entire fetus, and that cavity is known as the amniotic cavity. That is, above the ectoderm, amniogenic cells will form, the cells of the ectoderm will get differentiated and it will become amniogenic cells, and these amniogenic cells will secrete a fluid known as the amniotic fluid. Eventually later it will cover the entire embryo, that is the amniotic cavity. And next is decidua, that is endometrium during pregnancy we are calling as decidua. We know that the uterus is having three layers, endometrium, myometrium and perimetrium, and this endometrium is the inner layer where the fetus is getting implanted, and once after the implantation has occurred, that is once if the pregnancy has started, this endometrium will be renamed as decidua. It is a soft, vascular, spongy bed which allows the baby to get implanted. This endometrium is having three layers, basal layer, that is just the layer which lies above the myometrium, and once after the separation of the placenta, it helps for the regeneration of the endometrium in the puerperium. That is, after puerperium means it's a period after the delivery of the baby. After the delivery of the baby, when the placenta is getting separated, regeneration or restructuring of the endometrium should be there, and that will be happening due to with the help of the basal layer of the decidua. Next is functional layer. It contains secretory glands and it prevents the invasion of syncytial trophoblast and makes the separation of placenta easy. Syncytial trophoblast, what is that? I will explain you later. Actually, in one sentence I will tell, it's a layer of placenta. Okay, so the placenta is getting implanted, or it is getting growing into the uterus. Already I have told you that the type of implantation in the human being is interstitial implantation. That is, the placenta and other structures are growing into the endometrium of the uterus. Actually, this placenta should get restricted inside the endometrium. It is not supposed to go to the myometrium or perimetrium or to the deeper layers of the uterus. In case if it is going to the deeper layers of the uterus, after the delivery of the baby, when the placenta is getting separated, the separation of the placenta won't be easy if it is going to the deeper layers. If it is only in the superficial layer, it will be very easy for the placenta to get separated. So actually it is a responsibility of the functional layer to prevent the further inversion of the placenta to the myometrium or to the deeper layers of the uterus. Okay, and if it is not growing into the deeper layers after the delivery of the baby, when the delivery of the placenta is occurring, it will be easy for the separation of the placenta. And next is compact layer, and it contains, that is, we were telling that this endometrium during pregnancy is having 3 layers: basal layer, functional layer, and compact layer. This compact layer is the inner layer, or the layer which is coming close to the baby. This is having 3 layers again: basal decidua, capsular decidua, and parietal decidua. Basal decidua means it's a decidua which is coming under the blastocyst. Capsular decidua means it is the decidua which covers the fetus. Parietal decidua means it is a layer between the uterine cavity and the decidua basalis on the opposite side. Finally, later when the fetus is growing, this capsular decidua meets and fuses with the parietal decidua. I will explain it with the help of a picture. See, here you can see this is the fetus and this is the placenta. And if I am just explaining it with the help of a picture, means it will be like this. See, assume that this is a uterine cavity. That is the endometrium. And during implantation, what will happen means the fetus will come, it will get attached and it will erode the layers. See, actually the endometrium is thick. I'll make it as thick. Okay, this is the endometrium, because we know that it's having 3 layers. Okay, like this, full it is thick, up to here it is thicker. Okay, fully it will be there, assume. And assume that the fetus is coming and getting attached over here. What will happen means it will just start to erode and it will grow into the endometrium. It will grow into the endometrium. Okay, it will be growing inside and it will be coming like, when it is growing, it will grow towards the uterine cavity. This is a fetus. Assume that this is a fetus. Okay. And this is the placenta. This portion is the fetus. The structure is there. Here you can see this. Okay. And this structure is the endometrium, we know. And this structure, when the fetus is getting entered into this, actually some portion, this endometrium will cover this area of the fetus and it will be there here. And here also endometrium is there. We know that this is also endometrium. This portion is endometrium and the down portion is endometrium, and the portion when it is getting entered inside, this top portion also will be covered by the endometrium. Okay. And the portion where the baby is attached, this portion, that is known as the decidua basalis. Okay. And the portion of the decidua which is covering the baby, see here you can see a layer that is known as the decidua capsularis. And the portion of the decidua, here also endometrium is there. Only this portion is encroached by the baby, but here normal endometrium is there. And this normal endometrium is the decidua parietalis. So decidua basalis, capsularis and parietalis. Later what will happen means when the fetus is growing, when it is growing, this capsularis will come down. Further again and again the baby will push the capsularis downward. Assume this is the decidua capsularis. The decidua capsularis. When the baby is growing, this capsularis will pull downward. The baby will be coming like this downward, and it will finally merge with the balance endometrium. Okay, these 2 layers will come together. Decidua capsularis and decidua basalis will come together. Okay. Next is development of the placenta. Already I have told you that the trophoblast, you may be remembering the outer layer of the inner cell mass, it will develop the extra embryonic mesoderm. That is, already I told you that it will develop into the chorion and the placenta. This trophoblast, it will develop the extra embryonic mesoderm, and that is known as a chorion. And later small Villi-like projections will develop in the trophoblast. See, if you are just thinking about that picture was almost like this. A layer of cell was there, outer layer of cell was there, and here inner cell mass was there. Okay, and this outer layer we were calling as the trophoblast. This trophoblast is outer layer. This is the inner cell mass. Already we have seen the previous picture. And later what will happen means small Villi-like projections will develop in this trophoblast. Villi near the capsular decidua will degenerate, and this part is known as the chorion leavae. See, assume that differentiated structures I have just drawn as a single layer, and certain Villi-like projections will develop all over. These Villi-like projections are there. This is the trophoblast, and this is the Villi-like projections. And here we know that uterine endometrium is this is a uterine endometrium, and this is the basal decidua, and this is the capsular decidua. Already we have seen. Okay, so the villi near to the capsular decidua will disappear. See, and parietal decidua will disappear. Oh, sorry, the fetus will be there. Okay, here the villi has disappeared. Here villi are not there, see, and only the villi are there here. So the villi near to the capsular decidua, it has degenerated. Now this area is smooth without any villi, and this area is known as chorion leavae, and the villi near to the decidua basalis is still having that projection, and this area is known as a chorion frondosum. This is chorion frondosum, this is chorion leavae. And later a single layer of trophoblast become two layered. That is, it will become syncytial trophoblast and cytotrophoblast. See, this Villi-like projection and that layer is a single layer. Already in the previous picture I have shown that it's only a single layer. It was like this. Only a single layer was there. Okay, it was like this, and only single layer in the inner cell mass was there inside. Okay, it was outer thing only was a single layer. Later what will happen means this is only a single layer. Later this will become double layer. Okay, this will become double layer. Two layers will form. One more layer. Okay, so one layer. This is one layer, and this is another layer. The cells which are towards the baby, that cells will be with the cell membrane, and that is known as a cytotrophoblast. This outer cells, this is known as a syncytial trophoblast. These cells which are near to the decidua or to the endometrium, and these cells later it will lose the cell membrane. The cell membrane will not be there, it will disappear, and only inner cells, the cells of cytotrophoblast only will be having the cell membrane. So these cells without cell membrane, if you are looking, it will be there will be certain nucleuses, cytoplasm will be there, and all this nucleus and cytoplasm will be together, and it won't be having any cell membrane. That is the second layer. So now the syncytial trophoblast is there without cell membrane, only certain cell structures are lying without any cell membrane. Here cytotrophoblast is there, that is with fixed cell membrane. Okay, so now two layers are clear. Later what will happen means, I told you that one layer is there without any cell membrane, so it will be, what to say, one layer, all the other structures will be there. It's not having any cell membrane, nothing is there, certain cytoplasm is there, nucleus is there, that's all. It's not having cell membrane, so all these things are clumped together. Inside that we are having that clearly differentiated syncytial trophoblast with the cell membrane. Okay, now this is a structure. Later what will happen means certain cavities will develop inside this syncytium. See, this is the syncytial trophoblast, this is the cytotrophoblast. Later what will happen means certain cavities will develop in the syncytium. Oh, sorry. Say certain cavities will come. So what will happen means it will be up to here, it will be there. Later again it will be there. Again here it will be there. So certain cavities have developed. See, this is a syncytium and certain cavities are developed in between. And these cavities are known as lacunae. Cavities we are calling lacunae. And the cells of syncytium, this is also syncytial trophoblast, this is also syncytial trophoblast. We know that only one thing happened, it has got certain cavities, and the cells in between the syncytium, in between the lacunae, we are calling as trabaculae. This is trabaculae and this is lacunae. Later what will happen means this syncytium will grow into the endometrium. Again it will grow towards the endometrium. Okay, it will enter into the endometrium. Here we know that endometrium is a maternal blood vessels are there. So it will start to erode the maternal blood vessels. It will just come and erode damage the maternal blood vessels. So when the blood vessels are getting damaged, what will happen means blood will start to leak into this space. The blood will get filled into this lacuna space. From this maternal blood vessel, blood will start to leak into this lacuna space. So now trabaculae is there, lacuna is there, here maternal blood vessels are there, and here this lacuna is filled with the blood. And the cytotrophoblast grow into the trabaculae. This stage, the lacunae is known as the intervillous space. So in the previous picture, once again I will draw it here. This is in situ trophoblast. Again, this is in situ trophoblast. Again, it's in situ trophoblast. That is, now we are not calling it in situ trophoblast, we are calling it as trabaculae. This is lacunae. Lacunae, this is there with the maternal blood, and here we know that cytotrophoblast is there. Okay, cytotrophoblast is there, and this in situ trophoblast is thicker like this. Later, the cytotrophoblast, I will just make it with a different color. Cytotrophoblast, this is cytotrophoblast, and the yellow is in situ trophoblast. Okay, this cytotrophoblast will start to grow into the syncytium. This will start to grow upward. Okay, now when this here also it will grow. This is in situ trabaculae. This is also trabaculae. Here also this cytotrophoblast will grow. Here also the cytotrophoblast will grow. And now here we know that maternal blood vessels are there. Maternal blood vessels are there. This is cytotropoblastic layer which has grown into the trabacula or into the syncytial trophoblast. In this stage, this lacunae we are calling as intervillous space. Okay, this is intervillous space, and this is trabaculae, and inside that it is layer of cytotrophoblast. And this is a structure I have drawn over there. This mesoderm, in later what will happen means here, already here one cytotropoblastic layer is there. I have told you that cytotrophoblast also will grow into the syncytium. Later what will happen means down to that cytotrophoblast we know that mesoderm is there. This mesoderm also will start to grow into this syncytial trophoblast. Later what will happen means the vessels, the fetal blood vessels also will, fetal blood vessels from here, that also will start to grow into this syncytial trophoblast. So here uterine blood vessels are there, it has got eroded, and it is eroded and it is pouring blood into this intervillous space. Now this cavity is filled with the maternal blood, and here our cytotrophoblast, syncytial trophoblast outer it is there, it is in blue color. Then again cytotrophoblast one layer is there, again inside the trophoblast mesoderm also has grown inside, and into the mesoderm fetal blood vessels are also grown. And here now this complete structure, one structure we are calling as one villi. Here one villi is there, here one villi is there, like that. The prime villas are there. Inside the villas, intervillous space is also there. And here is the exchange of products which are happening between the fetus and the mother. Actually, there is no direct contact between the fetal blood vessels and maternal blood vessels. Actually, there are four barriers, four membranes which are acting as like barriers or which are filtering the maternal blood, and it prevents the direct contact with the fetal blood and the maternal blood. Fetal blood is inside this fetal blood vessels. So the layers are, first we know that outer syncytial trophoblast is there, inside that cytotrophoblast is there, second, next mesoderm layer is there, third, and fourth, blood vessels are having a covering membrane, that is a fourth layer. These four layers are separating the maternal blood from the fetal blood, and this is the placental barrier. And if you are looking into the placental circulation, the fetal blood vessels with less amount of oxygen, you can just remember the picture, the fetal blood vessels are entering into the villus, and these vessels will be with less amount of oxygen and less amount of nutrients. It will enter over there through the umbilical artery, and it will come to the placenta. It will enter into the villi, it will enter into the capillaries of the chorionic villi. There it will leave the carbon dioxide and it will absorb oxygen and other nutrients from the maternal blood. That is exchange of carbon dioxide, oxygen and other nutrients is happening over there inside the villus. See, if you are just looking into the structure of placenta, it will be like this. This is the umbilical cord which is coming from the baby. It contains umbilical vein and umbilical artery. Two arteries are there and one bluish color vein is there. This umbilical cord and umbilical vein is coming through this way and it is entering into the villi. Umbilical vein and umbilical arteries are there. It is there inside the villi, and this mesoderm is there. Mesoderm is also has gone into the villi, and here each villi, primary villi, each villis are there. In between the villi, intervillous space is there. Already we have seen all these things. And in the intervillous space, here uterine endometrium is there, and here the maternal blood vessels are there. This maternal blood vessels are pouring blood into the intervillous space. Okay, now let us see the functions of the placenta. It includes respiration. That is, respiration means it's the exchange of oxygen and carbon dioxide. Already we have seen that it is happening in the placenta. Next is it provides nutrients. Whatever the nutrients and minerals, vitamins which is needed for the baby is coming through the placenta. That is from the maternal blood through the intervillous space. It is getting entered or absorbed into the fetal circulation. And next is storage. It is storing certain amount of nutrients and immunoglobulins. Next one is excretion. That is all the waste products, carbon dioxide, urea, uric acid and all the waste products are getting excreted through the fetal placenta. That is from the umbilical cord. It will through the arteries. It will come to the placenta, and from the placenta it will get transferred into the maternal blood. And another one is protection. It prevents the invasion of bacteria except Trypanosoma syphilis and tubercle bacilli, and it gives IgG to the fetus. That is already I have told you about the placental barriers. 3 layers of cells are separating the maternal blood and the fetal blood. So these 4 layers are separating. These 4 layers will filter almost all the microorganisms except the microorganism of syphilis and tuberculosis, and it will pass the viruses unfortunately, and it gives immunoglobulins also to the fetus. And next is endocrine function of the placenta. It produces human chorionic gonadotrophic hormone, that is HCG, human chorionic gonadotropic hormone. It stimulates the growth and activity of the corpus luteum during pregnancy, and this corpus luteum It helps for the maintenance of the pregnancy, and the peak level of HCG, human chorionic gonadotrophic hormone, you can see between 7 to 10 weeks of pregnancy, and gradually towards the end of the pregnancy it will start to decrease, and it produces estrogen and progesterone also, and it produces another main hormone, that is human placental lactogen, HPL, human placental lactogen. It helps in the glucose metabolism in the fetus, because in the fetus we know that in the initial stage the pancreas may not be functioning effectively, so insulin production may not be effective. So this human placental lactogen, it will help for the glucose metabolism in the baby. This is the placenta structure. Once again I will explain it. These are the umbilical arteries, 2 arteries and 1 vein is coming to the placenta through the umbilicus, to the umbilical cord, and here it is entering into the villi, each primary villi. These are the villis, 1 villi, 2 villi, and blood vessels are there. Mesoderm, cytotrophoblast is there, syncytial trophoblast is there. Okay, all these cells are things are together, it is forming the villi, and here the endoderm, that is endometrium is there, Decidua, that is endometrium, it is there, and here myometrium is there. Actually the growth of the placenta is restricted in the endometrium, it is not going into the myometrium. You can see that myometrium is free, and it will erode. Here maternal blood vessels are there, maternal arteries are, and veins are there, arteries are red and veins are blue, and this will erode, this placenta is eroding or erasing the blood vessels or producing damage to the blood vessels, and it will start to leak, or it will start to pour blood into the space. Okay, this is the placenta. And if you are looking into the placenta, the total diameter of the placenta is 20 centimeter and its thickness is 2.5 centimeter, and at term, that is at the time of delivery, if you are taking the weight of the placenta, it will be 1/6 of baby's birth weight. That is, if the baby's weight is 3 kilogram, it will be 600 grams, or around 500 to 600 grams. And it is having a maternal surface and a fetal surface. The maternal surface, the surface which is coming towards the endometrium of the uterus, that is dark red in color, and it is having 20 lobes, each primary villi and its associated structure that forms a lobe. Like that, 20 lobes are there in each placenta, and each lobe is separated by sulci. And another surface is fetal surface. The fetal surface is the surface which is coming in contact with the fetal side, that is towards the amniotic cavity. It is covered by amnion, and it gives a white shiny appearance. This amniotic membrane, it gives a white shiny appearance to the fetal surface. So that's all about the basic development of fertilized ovum and development of the placenta, and we have seen the functions of placenta also. Thank you for watching this video. See you soon with the next video, and in that video we will be explaining in detail about the umbilical cord, about the amniotic fluid, how it is getting formed, all those things we can see in the next video. So wait for the next video.